Hello, good morning. Let's get started. We are doing lecture 5 today. So let's begin with some general review of some of the key things that you should know for this particular lesson. So lecture 5 is about the nervous system, part 1 of it anyways, and it deals more on the molecular level, how signal is being transmitted from one neuron to another, and signal being passed down the axon within a particular neuron. And next week, well, next week is engagement week, but then like next lesson, we will be doing nervous system part 2, which is more about the anatomy, okay, the structure of the brain, different regions of the brain, and their functions. So that's more like from a gross level, and today it's more on a molecular level. Okay, so when it comes to nervous tissue, there will be the neurons, right? Neurons. And then there are also going to be cells that will be supporting the neurons. Does anybody know the name of the supporting cells that I'm talking about? What do we call the cells that supports the neuron? That's the question. Anybody? Yes, thank you, Isabel. That is the glial cells. Yeah, glial cells. And Patrick is also right. They are also called neuroglia. Okay, so there are different types of glial cells, different types of neuroglia. I'll just give you some examples. We have things like astrocytes. Okay, and the function of astrocyte is to form what we call the blood-brain barrier. Okay, we will talk more about it next time. We have things like ependymal cells. Okay, so the ependymal cells, they would help form cerebrospinal fluid. All right, and again, we'll go into more details next week about these things. And we also have what we call like Schwann cells. Schwann cells. Okay, and these one, they would form the myelin. Okay, the myelin, which speeds up the conduction in the axon. If you put a layer of myelin around it, it is going to be able to conduct it a little bit faster. Okay, so those are the glial cells. Now, glial cells are basically there to support and to protect the neurons themselves. Okay, they are the ones that can divide, whereas the neurons they don't really divide at all. Okay, the numbers you have are pretty much going to be constant. They can form new connections, but if you lose some neurons, they're not really going to come back. Okay, so let's talk about the general structure of a neuron. And again, I've talked about this already in the recordings, but it's a good idea to just kind of refresh your memory. So there's going to be like a body, right? Cell body, and there's going to be like a really long thing that's coming out. Okay, so there's a nucleus. Okay. Now, coming out from the cell body, we have all these little projections. Okay, anybody know what these are called? These little things that are sticking out from the cell body. What do we call that? Anybody? You can look it up too, right? Like if you have it. Yeah. Shamira said dendrite. That's correct. Dendrite. And the function of a dendrite is to receive signal, okay, from another neuron usually, but it also could be from a receptor directly, right? A receptor is something that detects a change in the environment, Okay, so those signals will pass on to this particular neuron through the dendrite. Okay, now the signals that are coming in could be like red signals. Okay, so we call these inhibitory signals. Inhibitory signals. Okay, so basically, if you have a lot of inhibitory signals, then it's telling the neuron to, not do something. Okay, do not pass on the message. And at the same time, we can also have these green signals. Okay, green signals coming in. Okay, and opposite of inhibitory is excitatory. Okay, so a combination of these signals can simultaneously be stimulating the neuron, and then the neuron will do what we call like synaptic summation. Like they would add up all these signals and see which one is stronger. If there are more green signals, then you will pass on the signal. If there's more red signal, then you would, not pass on the signal. Of course, this is a simplification of what really goes on. It's possible that sometimes, even if you have inhibitory signal, you will pass that signal on to the next neuron, but you will be passing on like the inhibitory signal, right? Like you don't necessarily have to stop the signal here. You can be telling another neuron to stop sending a signal. Okay, so but just to keep it simple, if you have excitatory, it's go. If you have too much inhibitory, it's a stop. All right. What about this long thing right here? What is that thing called? What is that called? Anybody? That is the axon. That's right. Very good. Okay, so the axon is responsible for propagating the signal all the way to the axon terminal. Okay, and the axon could sometimes be insulated, right? Okay, so we have these little waxy coating, and they are called myelin. Now, depending on whether you are in the PNS or if you're in the CNS, the cell that forms the myelin is going to be different. So for example, we talk about Schwann cells already. Schwann cells are responsible for forming myelin in the PNS, the peripheral nervous system. Okay, you would have a different types of cells that would form the myelin in the CNS. Maybe I should write it down here as well. It's called the oligodendrocyte. Okay, so that would form myelin in CNS. Can somebody tell me the difference between CNS and PNS in a very simple term? Like, what's the difference? What's in the CNS? What are the two main things that makes up the CNS? No, nobody? Oh, thank you, Shamira. This is brain plus spinal cord. Brain plus spinal cord. Okay, so anything that is not brain, not spinal cord, that's part of the PNS. Okay, so like I have a nerve here, right? And like if I tap on the hand, I can feel that, right? So there's a nerve that sends a signal to my brain and spinal cord. That nerve that runs along my arm, that's part of the PNS. Okay, you have a nerve that on your face that allows you to feel like, cold fingers or warm fingers, right? That is part of your PNS as well. Anything that's not the brain, not the spinal cord, that is the PNS. Okay, so in the CNS, in the brain and the spinal cord, if you have a neuron that needs myelin, that's going to be produced by the oligodendrocytes. Okay, in the PNS, we would have the Schwann cells. Any questions so far? Now, this is a very difficult lecture because it's very abstract. So make sure you watch the recordings if you haven't already. Now, you don't myelate the entire axon. You need to leave a little bit of gaps. The gaps allows the signal to jump, okay? Jump like that. And these gaps, they are called the node of Renvier. Okay, it allows ions to be exchanged. Okay, and the exchange of ions at these locations, it's what's creating the signal that the neuron is passing on. Okay, you can think of the signal as like current, right? And you know, ions of course carry charges, and by exchanging ions at these locations, you allow the signal or the impulse to jump from node to node. Okay, and that's much faster than going through an axon that does not have myelin. Okay, so just in case you're not getting all these, myelin allows faster conduction. Okay. You might have heard of multiple sclerosis, right? Okay. In multiple sclerosis, your T cells attack the myelin, okay, and then without the myelin, you cannot conduct the signals, and it could lead to paralysis, for example, and death. So there you have it. Now, does all neuron look like this? No, not really. Okay, this is just like a typical one. You can also have neurons that are what we call like bipolar. Bipolar means rather than having, you know, one axon, you can have like two axons. So one axon here, and then there's going to be like another axon right there. Okay, so this would be a bipolar neuron. Okay. Now we also talked about the pathway of information flow, right? Which is basically like this diagram. Okay, I fill in this entire page in the lecture recordings. So you can watch that again if you haven't already. But I'm gonna do like a quick overview here for you. Okay, so flow of information. Flow of information. Okay, so you know, this is your hand. Okay, and let's say there's something sharp that pokes you. There's a sharp pin or something that pokes you. Okay, so what happens is you are going to have in your skin, in your epidermis, you are going to have a receptor. Okay, so the receptor looks something like this. Okay, this would be a sensory receptor. Sensory receptor. So you have different types of sensory receptor throughout the body. Okay, you have sensory receptor that detects light in the eye. You have sensory receptor that detects like taste on the tongue, and in the skin you have sensory receptors for pain, for temperature, for pressure. Right. So over here, if you're being pricked by a pin or something, then it would be like one of the pain receptors that picks it up. Okay, so sensory receptors detects a particular sense, if you would, okay, a particular change in the environment. And then you're gonna have one of these you know, special neurons, okay, that would pick up the signal, and then it will go all the way. This is like the cell body, and then it will take that signal towards your central nervous system. Okay, so this neuron that does that, that is the sensory neuron. Okay, sensory neuron. Okay, the job of the sensory neuron, let me say that one more time, is to take sensory information, takes sensory information towards the central nervous system. Does everybody understand what I mean when I say sensory information? Any question? All right. So here is a sensory neuron. Now, of course, there's not just like a single neuron because that's like super flimsy and it's very vulnerable. So you actually have a bunch of these things. But for simplicity, I'm just going to draw one. But know that there's going to be like a bunch of them, okay? Like a bunch of them like that. Okay. So these sensory neurons will form a bundle. Okay. So we call that the afferent pathway. Okay, the afferent pathway takes information from the PNS towards the CNS. Okay, this afferent pathway is made up of sensory neurons. And what does it carry? It carries sensory information. In this case, pain and pressure. All right. The same thing's going to happen. This could be your eyeballs. Right? If it's your eyeballs, then the sensory receptor will be detecting light. And then that information will be also carried through the sensory neuron. Okay. But then the type of information would be like light. Okay. Then it's going to go to your central nervous system. Okay. Where you're going to have another bipolar guy right here. Okay. So this is an interneuron. Interneurons are special because they are only found in the CNS, only in the brain or the spinal cord. Okay, it does the thinking. So what it does is it will integrate the sensory information. Okay, this is very simple pathway, right? Just one pieces of information coming in. Okay, but there could be multiple information coming in. Like think about like if you're walking on the street, okay? So many different senses is coming in. Okay, you have to see where you're going, right? Your hearing helps you detect if there's any, I don't know, like a car coming towards you or something. And then when you're walking, you have to shift your balance left and right. All that information are processed by the interneuron, okay, within your central nervous system. And then it's going to decide what to do with it, okay, what to do with that information. And if an action is required, such as this case, okay, if you get pricked by something sharp, you're gonna remove your finger, right? So that is gonna send out through this pink neuron. Okay, anybody know what this neuron is called? The blue one is the sensory neuron. What's the pink one? Please help me out here.
Motor nerves.
That is correct. That is the motor neuron. Okay, motor neuron. Very good. Okay, and what does it do? Well, it takes motor command towards the effector, right? The effector is the thing that's going to be carrying out the action. Okay, so in this case, the effector is going to be like the muscle in the hand. All right, and again, it's not just going to be one of these pain things. You're going to have multiples of these, and it's going to form the opposite of afferent is efferent. Okay. Some people say efferent, both is okay. But E for exit, that's how you can remember it, okay? It exits from the CNS, right? And then it travels towards the PNS. Right? Well, technically, it's traveling towards the effector. Okay. And so in the end, you will remove the hand. So this is the flow of information. Is that okay? Any question about this? Alrighty. Do we have some polls? Maybe later. Okay, we cannot do those yet. Okay, what's on the next page? Now this action potential thing, right? Okay. Do you guys want me to talk about this again? Like, let me just do a quick poll. And do you want me to go through that one? Like like do a summary like what I did here, or like you guys are okay? Like yes for A, B for no, thank you. Where's everybody? It's like only 3 or 4 people who's responding. Are you guys there? Hello, hello. Okay. All right. Everybody who voted said yes. And for the ones who are maybe not at their keyboard, I guess it doesn't really matter for them. All right, so let's do it. Let's do it. Okay. Now, to understand that whole action potential ordeal, like this graph, okay, I'm going to try to draw this graph out again for you, and then we're going to explain it, okay? Do ask questions if you are not following, okay? It's very important that you do. Can you show the previous page briefly? Yeah, sure. There you go. I'll post this as well, right? Of course. But, you know, go ahead, take a picture. Let me know when you're good to go. Yep. Actually, before the action potential, I forgot. We also have to talk about the division of the nervous system. Okay, so let's quickly talk about that as well. So here we have the nervous system. Okay. And as you know, the nervous system is divided into the central nervous system. Okay, the CNS, right, which again is going to be brain plus spinal cord. When you discuss this, are you going to fill out the chart? What chart? Like this chart over here? Yeah, sure. We could do that. Just remind me if I forget. Sorry, what was it talking about? Okay, the central nervous system, brain and spinal cord, right? And then we also gonna have the other one, right? The other one, as I said, would be the PNS, peripheral nervous system. PNS. Okay, so everything that's not the brain and the spinal cord, that's going to be part of your PNS. Okay, and if this is a little bit abstract for you, okay, let me just quickly show you. You don't have to draw this. This is just going to be like a really rough diagram, chicken scratch, as I say. This is your brain. Okay, and then this is the brain stem, right? Remember the medulla oblongata that we talked about in the respiratory system, right? Where you have the respiratory center. So that's part of the brain stem. And then this is going to come out from a hole at the bottom of your skull, and then it's going to go all the way down. So this whole thing, this is the spinal cord. Yes, spinal cord. Now I don't know if you know, but the spinal cord is going to become like ponytail down here. Okay, like that. Okay, so this whole thing that I draw here that looks like, you know, you pull an onion out of the ground and then you hold it upside down. That's what it looks like. This is your central nervous system. That's all there is. Brain plus spinal cord. Okay. Now, coming out from the brain, you have all these special nerves, right? Special nerves. These are called cranial nerves. Cranial nerves. And you have 12 pairs of them. 12 pairs of cranial nerves. Okay, those are not part of your central nervous system. Okay, it's out of the brain already. It's not part of the CNS. Okay, you also have nerves that are coming out. Okay, from your spinal cord. Okay, so these are spinal nerves. The only difference between spinal nerve and cranial nerve is where do they originate from. If they originate from the brain, then we call them cranial nerve. If they originate from the spinal cord, then we call them spinal nerves. How many pairs of spinal nerves do we have? Anybody remember? Thank you, Isabel. 31 pairs. That's correct. 31 pairs. Like I said, right? 12 months in a year, 31 days in some months. Okay, that's how I remember it. So there it is. Remember, these are part of the PNS. Okay. Now, in case you get a little bit confused about what a nerve is basically a bundle of axons in the PNS. Okay, a bundle of axons in the PNS. So hold on, let me go up here. You see this afferent pathway, efferent pathway, that would form like a sensory nerve, right? Because it's a bundle of axons in the PNS. Okay, I should probably put a division here, right? This is the CNS because that's where the interneuron is, and everything outside of that, that's the PNS. So if you have a bundle of axon, right, that is going to be a nerve. Okay. So now we are talking about the peripheral nervous system. Let me move this thing out of the way. It's kind of blocking me. Let's make it smaller, and then we'll move it over here. There we go. Okay, so we are talking about the PNS, right? The peripheral nervous system. Functionally speaking, you can separate the PNS into two other separate branches. So let's do that, okay? We can separate it into the autonomic nervous system. Okay, so we call this the ANS. Now you gotta know, like this thing is really complicated, right? The whole nervous system thing. So I'm just trying to simplify it as much as you can. So for our purpose, the autonomic nervous system is the branch of the PNS that is responsible for everything that's automatic within the body. Okay, so what's automatic within the body? Regulation of your heart rate, right? You know, changing the size of your pupil, you know, constriction of your blood vessels, all these things that happens without us consciously thinking about it, that is going to be the work of the ANS. Okay, like when you're sleeping, you will still continue to breathe, right? That's the job of the ANS. So everything that occurs in the body without conscious input, right? That just kind of happens in the background. That's the job of ANS. Does that make sense? Okay, not hearing anything. I assume that makes sense. So thank you, Yetiman, for saying yes. And then the other side is going to be the somatic nervous system. And this is going to be the SNS. So this deals with everything that is you know, voluntary, deliberate. Okay, like I'm talking right now because I choose to talk. So in order for me to talk, I have to use pathways within the somatic nervous system. I'm gesturing, I'm moving a hand. Okay, I can move my fingers at will. All those movements will be controlled through this particular division. All right. Now, under the autonomic system, nervous system, there is also going to be two additional systems. Okay, we have the sympathetic system, and we also have the parasympathetic system. Okay, so these two systems, they work opposite to each other. Okay, and the sympathetic system is most active when you are excited, when you are in danger. Okay, it's responsible for the fight or flight response. Okay, so it will do things like increase your heart rate, increase your breathing rate. It will divert more blood to your muscle so you can like run away from the danger, for example. It will dilate your pupil so you can see better, right? So all these things are going to be the function of the sympathetic system. On the other hand, the parasympathetic system is to calm you down. Okay, so like if you're just like resting and relaxing, very calm, then the parasympathetic system is going to be most active. Okay, so it will slow your heart rate, slow your breathing, constrict your pupil, promote digestion, all those kind of things. Okay, so these two things, they are antagonistic, opposite to each other. Okay, and for a list of what I just said, you can look at the PowerPoints. So any question about all these divisions that I was talking about? All right. Now I'm gonna do something that will combine what we learned here with what we have here. Okay. Try to stay with me, and hopefully it will not be too confusing. Okay, so let's say we have the same situation, like you touch something that's sharp, and then your hand moved out, right? So where does that begin in this entire pathway? Which division of our nervous system would the pricking, right? The sensation of being pricked, where does that originate? Which of these divisions? Does it begin in the CNS? Does it begin in the PNS? And if it begins in the PNS, like where? Anybody want to take a shot at that? The question is, when you prick your finger, which part of the which division of the central of the nervous system is responsible for that?
Is that PNS systematic system?
It is the PNS, very good. But which part of the PNS? Do you think it's ANS or SNS if you prick your finger?
ANS.
Okay. Anyone else? Isabel also think it's ANS. Yeah. Anyone else think otherwise? Okay. Jane said is SNS. Okay. Now here is what you do. Okay. Like on the test, how do you figure out which one is which? Right. You think about the body part that is being triggered. Okay. In this case, you prick your finger. Right. Do you normally have voluntary control over your finger or not? I hope you do. Okay. Like you can move the finger at will. Okay. And because you have voluntary control over it, is that ANS or is that SNS? Right? Think about that, right? It's going to be SNS, isn't it? Okay. So if you prick your finger, right, that afferent pathway is going to begin right here, and then it's going to go towards your central nervous system. Okay. So this is like pricking your finger. Okay. And again, it begins here because that's something you have voluntary control over, right? And then the central nervous system, the interneuron, the green one, right, will process that information. It'll be like, oh no, we touched something sharp. We should remove our finger. And then that's going to send the signal. I got to use that same color from above. It's going to send the signal back out, right? And then the effector is going to be your finger and you're going to move away. Make sense? All right, here's another one. You eat too much. Okay, you went to a buffet or something. All right? Pandemic is finally over, and then they open up the buffet. You go eat as much as you can, and then you go home and you realize you ate too much, and then your stomach is too full. It feels uncomfortable. Okay, so the pain is starting coming from the stomach. All right, is that ANS or is that SNS? Again, you think about, do you have voluntary control over your stomach or not, right? Okay, you do not. Okay, so Safi is correct. It is the ANS. So in that case, right, it is going to be coming from this particular division. Okay, and it will send the signal, right, to the CNS, and then I'm not sure what can be done here, but like it will send signal back to the stomach, or like it will send signal to other parts of the body that makes you feel like that uncomfortable, that uncomfortable feeling that you get is basically the consequences. Okay, so this would be like I said, you know, let's just put like another example, like kidney stones or something. Okay, so that stretches the kidney, and then you have a receptor for pain, and then that will be processed through the ANS. Does that make sense? I hope so. I hope so. All right, let's do some polls. Thank you, Mary, for confirming. There we go. This is a multi-select question. Okay, all these questions I'm gonna do with you today, right? All these are like taken from the bank, right? So hopefully it will help you a little bit. Go ahead. All right, I have 3 people so far, 5 now. Just gonna give it another, 30 seconds or so. Okay, very good. All of you have answered. Wow, for once, everybody did the polls. I'm so proud. Very good. Okay, so what's the answer, right? You're looking for PNS. Okay, so anything that does not say spinal cord or brain, right? We cross out the brain, we cross out the spinal cord. Everything else is good. Okay. Nobody chose the spinal cord and nobody chose the brain. Excellent. Okay, everything else is the answer. Good, good, good. Okay, next one. This is just multiple choice. Oops. Go ahead. I don't think I have explicitly mentioned this yet, but it's okay. I'll tell you about it afterwards. Sorry. Okay, so nobody got this one, but that's okay. I mean, it was talked about in the lecture, but I didn't talk about it today yet. So maybe that's why you guys don't know. So nerve cell, this is one nerve cell, right? Oh, why is the pen so thick? There is the neuron. Okay. And then the impulse. Impulse means signal, okay? Signal goes from one nerve cell to another nerve cell. Okay, how does the signal go from this to this? Where they are going to be passing the signal, that is called a synapse. Okay, synapse. So that's the answer. More on synaptic connections later, but that's the answer. Okay, I wrote this down, see if you remember. If you don't, hopefully you know where to find the answer. Okay, well, I mean, it's good that we are down to two choices for most people. And for the central nervous system, it is going to be the oligodendrocytes. Okay, so this is the CNS and this is the PNS. Okay, CNS is oligodendrocytes, little star-shaped cell. That's what it means. Schwann cells, probably named after the guy who discovered them. Yeah, so the answer is the first one. All right. If you didn't know, now you do. Yeah, the quiz for this one is coming after the engagement week. Okay, I believe. So no class next week. Right? Next week is week off, and then when you return, you will have the quiz. Okay, so that's that. Let me see if I missed anything else. No, I think we're good. Now we're gonna do the action potential stuff. All right. So please grab some markers and we will draw this together. Okay. You have to draw it out together to help make sense of it. Okay. Otherwise, it's a very passive experience. All right. Okay. So again, I'm not going to go into the full details that I did in the lecture. I'm just going to give you like a condensed version of it. Okay. It's like those YouTube videos where they like tell you the whole movie in like 10 minutes or something, right? So this is kind of like that. Okay, so this is the axon, okay, the axon. And this would be like the cytoplasm. And then this would be the outside. Right? Outside is extracellular fluid. Outside. Okay. Now, the first thing you got to know, first thing you got to understand here is that along the axon, there is going to be a lot of membrane proteins. Okay, so we'll be looking at three major ones. We have these sodium potassium pump. Okay, there we go. Sodium potassium pump. Can anybody please tell me what is the function of the sodium potassium pump? What do they do?
Transfer sodium out of the cell.
And that's correct. They will pump sodium out of the cell. And what does it bring in?
Potassium.
That's right. Okay, so it will use ATP to pump 3 sodium out in exchange for 2 potassium in. Okay, so we're going to be pumping out 3 sodium, pumping it out, and in exchange, we will be bringing in 2 potassium. Like that. All right, so I hope you got your markers. So if you bring a lot of potassium in, you will end up having a lot of potassium on the inside, right? Okay. There we go. And then not so much outside. Okay. And then for sodium, it is the other way around. You will have a lot of sodium. Okay. This is like using your bingo stamp. Like that. And then not so much. Okay, for those of you who do not like stamping all around, we'll put it in words. On the outside is high sodium. On the inside is low sodium. Okay, and for potassium, it is the other way around. Low potassium on the outside, high potassium on the inside. Okay. Any question? Hope it makes sense. Now, if you don't have questions, then I have a question for you. Okay. After this goes on for a while, is the outside more negative or is the inside more negative? Let's do a poll and see. Okay, the question is, after you let the sodium potassium pump runs for a while, is the outside more negative? Outside more negative? Or is the inside more negative? What's it gonna be? All right, let's see. Anyone else? There we go. Okay, so the correct answer is the inside will be more negative. And why is that the case? Well, let's think about this. Okay, we are pumping 3 positive out, but 2 positive in, right? So if you do a little bit of math, every time this completes a cycle, you are pumping 1 positive out, right? Basically, right? Because you're taking 3 positive out, but then you're bringing 2 in. So, you know, overall, you are pumping a positive out, right? So if you keep on doing it, then the outside will become more positive, right? And if the outside is more positive, right? Another way to say that is basically the inside is more negative, right? Okay, so the people who chose this one, have I convinced you that the inside is more negative? You have to understand why the inside is negative. Otherwise, nothing else would make sense from this point on. Anybody need clarification? You can just private message me if you don't want people to know who you are. Okay, all right. So the function of the sodium potassium pump is this, and then in terms of what we're talking about, what ends up happening is it established here's a special word, resting potential of a neuron. Okay, so what is a potential, right? A potential is basically like a charge difference between 2 points. Okay, so in this case, we talk about the membrane. So the inside and outside, it's not the same amount of charge. Outside small positive, inside small negative. So we call this the potential of a neuron. Okay, and right now the neuron is not doing anything. It's not transmitting a signal. It's at rest. So we call it a resting potential. So how do we get the resting potential? It's the work of the sodium potassium pump. All right, so establish the resting potential of a neuron. Okay, which is how much? Okay, anybody have anybody remember the number that I used in class? What is the resting potential? What number is it going to be? If I'm going to measure it, if I stick a probe on the inside and stick a probe on the outside, and then I read it on a voltmeter, what would the number be? Anybody? That's right. That's going to be minus 70 millivolts. All right. That's the resting potential. Okay. All right. So that's that. Now, on the membrane, we also have other stuff. Okay. So let's start a new diagram. On the membrane, we also have other stuff. Okay, so we're going to draw a new diagram. Okay, we have two other types of channels. Okay, we have the sodium channels. Okay, and then we have the potassium channels. Okay, now I'm not going to draw the sodium potassium pump anymore, so that it's not going to be too confusing. So there is the potassium channels. Okay, so potassium channels. We call them voltage sensitive potassium channels, or we call them like gated channels. Okay, because they can open or close. So the potassium channels, they open at positive 30 millivolts. And I should say that I round these numbers from like the textbook and online stuff so that it's easier for you to remember. So don't be surprised if you see something a little bit different. And they will close at minus 90. Okay. And then we have the sodium ones. Okay. So these are the gated sodium channels. Okay. They would open at minus 60, and then they would close at positive 30. Okay, it's just the way it is. There's no reason for them. People did some experiment and they found out that's what it is. Okay. So normally they're close, right? At resting, we are sitting at minus 70. So nothing is open. Nothing can pass through. And just let me refresh your memory. On the outside, we have high sodium. And on the inside, we have low sodium. And it's the opposite for the potassium. Okay. Now, another important question. Does the sodium want to come back in, or does the sodium want to go back out? What do you think? In or out of the cell? Let's do another poll. Okay. The question is, when given the opportunity, Does sodium want to A, come into the cell, or B, go out? What does it want to do? Does it want to come in or does it want to go out? Remember, you have a lot of these sodium outside. A lot of it. My chicken pox. There we go. And then I'll do. Okay. So when given the opportunity, where do they want to go? Okay, most people got it right. Yeah, they want to come in. Okay, because things naturally go from high concentration to low concentration, right? They cannot come in yet because the sodium channels are all closed at this point, right? Okay, and as an analogy, these are like people who are lining up on Black Friday and waiting for the doors to open. As soon as the doors open, they will swarm in, right? All the sodium will go into the cell. Does that make sense? I hope it does. Okay, now for the potassium, it is the other way around, right? Okay, I'm not going to draw the potassium, but the potassium is going to be high on the inside and low on the outside. Okay, so they really want to get out. Okay, they are like the Walmart employee that has been working all night just to get the store ready, and then they are ready to go out. Okay, but they can't go out yet because the doors are closed, right? The potassium channels are closed. The main point I'm trying to make here is, why is this dot not erased? There we go. The main point I'm trying to make here is the sodium will come in when the sodium channel is open, and the potassium will go out when the potassium channel opens. Okay, those are the 2 points that you have to remember. Why don't I write that out for you so that you will remember? What did I say? Sodium will enter the cell. Enter the cell when the sodium channels open. When will they open? When are they going to open? Well, they will open at minus 60, right? We're not there yet. We are at minus 70. We are at rest. Okay. And then the other one is the potassium. Potassium. Potassium will leave the cell when the potassium channels open. Any question about any of the stuff that I've talked about so far before it gets like even more confusing? Maybe it's not that confusing. Hopefully I've made it simple enough for you to understand. Okay, I will take your word for it. So, you know, on the side, if you have another piece of paper or something, okay, let's draw a graph to represent this. Okay, let's draw a graph to represent this. We are gonna have, okay, draw a big graph, okay, please. All right. Draw a nice one so you can stick it next to your computer when you do the test. Okay, if this is your piece of paper, I don't want you to draw the graph like this, okay? And turn the paper sideways and then draw a big graph. Okay, big graph like this. There we go. Okay, and then you stick that on the wall. Okay, so on the y-axis, we have our membrane potential. And membrane potential, again, it just means the charge difference on the inside versus outside. And we measure that in millivolts. Okay, if you look at like your battery, right, like your standard, you know, AA battery, it's like 1.5 volt each, right? It means between the positive terminal and the negative terminal, you know, your battery, right? Like one end is positive, one end is negative. It means over here, if you compare the charges stored here and the charge here, it's like 1.5 volt difference in charge, right? And the thing about charges is if there is a difference and you connect them with a wire, then the charge would move and it creates current, right? So instead of talking about the positive and the negative terminal, we're talking about the inside versus the outside of your membrane. Okay. Over here, this is time, and all this happens really quickly in terms of milliseconds. Okay. So we are at rest right now. We're not doing anything. Where should we begin on the y-axis? What number should we begin at on the y-axis? If you're thinking about -70, then you are absolutely correct. I'm going to write it right here. Okay, this is of course not to scale. So that's where we're going to be starting. Okay. Okay, I'm sorry, I'm going to be jumping all over the places. If this was done in a classroom, like we actually have 3 blackboards on the classroom. So it's very nice to be able to do all these things at the same time, but now we just have to jump back and forth. All right, so this is page 2 of the study guide, and this is the graph we are essentially creating. So at number one, what phase is this? What phase is this? This is the boring phase because nothing is happening. This is the resting potential. Okay, and we are sitting at -70 millivolts. Key events, nothing is happening, right? So we have low potassium, low sodium and high potassium inside the cell. Okay. And what creates this -70? Right, -70 MV is established by sodium potassium pump. Okay. Two potassium in, 3 sodium out for every cycle. Okay, let's take a quick break before we continue. And if you have questions, leave them in the chat and we'll get back to it. Okay, so let's resume in 5 minutes. I'll come back at 9:37. Okay, see you in a bit. Hello, it's me again. Let's keep going. So what's going to happen? That will trigger a signal to be transmitted. Okay. Let's go back to our diagram here. Okay. What's going to be happening is there will be some kind of stimulus that will arrive. Okay, so there's going to be like some kind of trigger, and I'll explain what that trigger means in a bit. But you can think of like your finger touching something sharp. That's going to create a trigger, right? A signal. So that trigger will travel. Will be represented by this lightning bolt, and that trigger is going to push the membrane potential a little bit higher. Okay, so if we go back to our drawing here, which is really big, let me just make it a little bit smaller. Okay, so if we go back to our drawing here, what's going to happen is there is going to be the stimulus, the lightning bolt. Okay, lightning bolt. Stimulus. Okay, and that will push our membrane potential a little bit higher. Okay, sometimes it will just go up a little bit and then falls back down if it's not a strong trigger. Remember, you're getting these positive and negative signals all at the same time. So sometimes the negative signal is really strong, and then you won't be able to go up to the level that we need. Okay, but in some cases it will go higher than this particular threshold. Okay, so this is -60 millivolts. Okay, this is what we call the threshold potential. Threshold potential. This is the point of no return. If you cross the threshold, then a signal will be transmitted for sure. Okay, so At minus 60 millivolts, if you remember, that is the opening potential for the sodium channels, right? So at this point, the sodium channels open. Okay, so let me just remind you about our condition so far. At the beginning, at the resting potential, we have low sodium on the inside, and we have high potassium. Okay, you should know this by now. I probably have said this more than 5-6 times by now. Okay, and like I said, the sodium channel during resting potential is not open. However, if a stimulus arrives and it triggers and it pushes the membrane potential over minus 60, then it will trigger the sodium channels to open. Okay, let's think back to what we said before the break. When you open the sodium channels, what will happen to the sodium? Which way will they go? Let's take a look at our diagram here. Right? Sodium will enter the cell when the sodium channels are open. Okay, so that means as soon as these things open up, okay, open up. Let's just draw like a double door or something. Open up like that. Then the sodium will start coming in. Okay. As the sodium comes in, it will cause the inside to become more positive, right? And that's why the graph is going to keep on going up, like that. Now, it's not going to go up forever. It's going to go up to about 30 millivolts. Okay. Oh my goodness. Give me a second. I'm just going to erase it. This is membrane potential. It's going to go up to about 30 millivolts because that is the closing voltage for our sodium channels, right? Okay. So this rising phase is caused by sodium influx. Influx just means coming into the cell, and we call this depolarization. Okay, it's going to go up like that. So let's go back to our study guide here. What's happening at number 2? At number 2, we have a stimulus arriving. So arrival of stimulus, a trigger, okay? And at this point, it is still sitting at minus 70 millivolts, okay? And like I said, it might go up and come back down if it's not strong enough. Sometimes if it's a lot of negative signal, we can push it further down, so it's not as easy to fire the neuron, okay? So key events, some sensory receptor is triggered. Okay. And then at number 3, right, that is what we call the threshold potential. Okay, and that is, of course, minus 60 millivolts. Okay. So the key event that happens at the threshold is as soon as you cross the threshold, the sodium channels open. Okay. And number 4 is referring to this entire segment here. Okay. That is called the depolarization. Okay. So polarize means it's more negative on the inside in our context. So to depolarize is to undo that negative. And it's going to be from negative 60 all the way to positive 30. And the key event that's happening here is the sodium influx. Okay, sodium influx. And then afterwards, what's going to happen is, at 30 millivolts, sodium channels close. And then potassium channels open, right? Because that's the opening voltage for the potassium. Questions? No? No questions? Okay. Everybody done copying? Anyone else still needs a bit of time? Okay. So let's take a look at the situation right now. Okay. Let me redraw this in a new diagram for you. So again, we have our axon. Okay. We have our sodium channels that are opened. Now you have to understand the axon is really, really, really long. So the sodium channels that are further down our axon, they're still closed. Okay, they're still closed. Okay. So sodiums are coming in from here, okay, but nothing's really going on further down yet. Okay. What about the potassium channels? They're still close. Right? We haven't reached our 30 yet. Okay. So they're still close. All right. So as the sodium is coming in, now the inside will have high sodium, yes? And the outside will have low sodium. What about potassium? What should I put for the concentration for potassium? Is potassium high or low relative to outside? Okay. We have two people who say low. High. Okay, let's do a vote. Why not? Okay, just to see where people are at. My question is, when the at this point, what should I put for the potassium here? Okay, A for high, B for low. Okay. Relative to the outside, is it high or is it low? All right, looks like we are half here. Well, let's take a look at before. Okay. Remember all those blue dots, right? The high potassium, they want to go out, right? They really want to go out, but they cannot. Why can't they go out? Well, the potassium cannot go out yet because the potassium channels are not open yet, right? So you see, as the sodiums are coming in, the potassium, they're not going anywhere because the potassium doors, the potassium channels are still closed. So because of that, it will still be high, just like before. They didn't have anywhere to go yet, okay? Can't go anywhere yet. because potassium channels are still closed. Does that make sense? That it's the same as before? Have I convinced you that it's not low? Okay, thank you. I love convincing people. Okay, so over here, right, at the very peak, at the very top here, at the 30, all right, I'm just gonna put some labels for you again. It is going to be high sodium now because of the influx, okay? High potassium because that was the way it was before. Okay, we didn't open any potassium channels yet. Okay, at the peak right here. All right, so 3 things happen at the top here. 3 things happen. Okay. Number one, sodium channels close. Number 2, potassium channels open. Okay, and then number 3, we'll talk about number 3 in a second. You know what? Let's talk about it now. I'll let you copy that down first. Then I'll talk about it. Okay, let's go back to our lovely drawing here. Let's look at what happens further down the axon. Okay, further down the axon. This is the direction of transmission. Direction of the impulse. Impulse is just fancy word for signal. Okay, so here we go again, right? What should I put for sodium over here? High or low? And what should I put for potassium? Okay. Let's focus on the sodium. What should I put for the sodium here? Is it high or is it low? Let's think about that one. Okay, Safi said low. Anyone else? Anybody agrees, disagree? Okay, we have a bunch of lows coming in, low, low, low. Yes, that is correct, right? Because these doors are not open yet, right? So the sodium cannot come in just yet, okay? So let's take a look at here with the numbers, all right? And I hope this makes sense. I know this is like very weird stuff, and you might be wondering why am I learning about this? I have no idea. The ministry said you have to learn it. Okay, so this is positive 30, and then this is minus 70. Okay, so look, the sodium coming in is high, and then the gauge will close at 30, right? So these sodium can't really go back out because the membrane is blocking it, and then the doors are closed. So where is it going to go? Well, if this is high and this is low, some of the sodium is going to go forward, right? Okay. So as the sodium goes forward, it of course carries a positive charge. Okay. So as the sodium goes forward, what's going to happen to this number? Well, this number will not be negative 70 anymore, right? Because of all the sodium coming forward. So that will go to maybe minus 68, minus 65, minus 62, and eventually it will push it all the way to minus 60 if enough positive goes forward, right? So that would then trigger these sodium channels to open, and then the action potential will occur over here, right? Like the sodium will come in, right? And then that's how the signal goes forward. All right, so the third thing that will happen in our lovely graph here is that sodium channel, even though the sodium channels of our current section will close. Sodium channels further down the axon will open, right? Allowing the signal to go forward. Okay. Excuse me. Okay. If everybody's happy with this, let's take a look at what happens to our graph next. Now, the potassium channels will open at 30, and as soon as the potassium channels are open, then our potassium will leave, right? They wanted to leave in the first place anyways. As the potassium leaves, you will become negative once again. Okay, we keep on dropping, and it will drop all the way to minus 90. Okay, so why is it like that? Right? Why is so much potassium leaving, right? Okay, so this here is called repolarization. Repolarization. And it is caused by potassium efflux. Right? They're leaving. Okay. So I'm going to put a star here. Star. Okay. Two driving force. Okay. Two driving forces to push the potassium out. Okay, so number one, concentration gradient. Okay, potassium was high on the inside, it's low on the outside, it wanted to leave. That's understandable. But why so much of it leaves to the point where we're going to get to negative 90? Second driving force is electrostatic repulsion. Okay, let's go back to here for a second. Remember, you have all the potassium here to begin with. Okay, and then the sodium comes in, and then it creates even more positive, right? So all these positive charges, they don't like to be next to each other, right? Like charges repel, right? So all these high positives will help push the potassium out as well. Okay, so these forces combined will cause a lot of the potassium to go out until we get to all the way to minus 90. Okay, so the potassium channels will close here. Potassium channels close. And at this point, it's way below, lower than our negative 70. So we call this point hyperpolarization. Okay. It's too negative compared to resting. Okay. All right, let me go back to this one over here. So at number 5, where's number 5? Number 5 is like this phase here, right? Okay, number 5 is repolarization. Okay, and it is from 30 to minus 90. And it is caused by potassium efflux. Okay. And number 6 here is hyperpolarization. At minus 90. Okay. And so at this point, we kind of have two problems. All right. Let me just go back here. Sorry, keep on going back and forth. Okay. So over here, we have two situations at hyperpolarization. First of all, let's do the sodium and potassium thing first. Okay. What's going on with the sodium at this point? Is it high or low? Is the sodium high or low at this point? Yeah, it's high, right? Okay. They all came in. They didn't have a chance to leave yet. Sure, some went forward, but it's not a significant amount, so it's still high. And the potassium is low because they all left, right? In the efflux, they all left, right? So at hyperpolarization, we have two problems. Okay, let me put it in the chart here for you. We have two problems. Okay, we are more negative than resting potential. Okay. That means we cannot transmit another signal until we go back to resting. And then second problem is ion distribution is opposite compared to resting. Okay, so how do we fix these problems? We need the sodium potassium pump. We'll reset. The conditions. Okay. So the sodium potassium pump will take all these extra sodium that we have, put it back out, bring back some potassium, and then it will reset it to -70. And we're good to go again. All right. So there you have it. All right. Just gonna go back up to here. Now this temporary reversal of membrane potential, that's what we call the action potential. Okay, so this here, that is the action potential. Okay, it's a temporary reversal of membrane potential. Okay, so when you're studying for this, you should start with a blank piece of paper, and then you should draw your own graph and fill in the information as much as you can. All right, that way it will help you remember what happens where. Are we okay? Any questions? Okay, so let's try to understand this in grand scheme of things, as they say. So let's say we have the neuron. You don't have to draw this; you can just watch. Okay. So then we have a trigger, okay, stimulus that arrives. Okay, let's say we have more green signals than red signals. Then, it will push the first portion towards the threshold, -60. And then the rest of it will still be at -70, right? Okay. Then what happens next? Okay. This is a play-by-play action. Right? So -60 is going to open the sodium channels. All the sodium is going to come in, influx, and then that's going to push it all the way to 30, right? Okay. With all the sodium coming in, and then the sodium channels will close, some of the sodium will move forward, and then we'll push the next section to -60. And then the rest of it is still -70. Okay, so the influx of sodium causes the membrane potential to flip in this section. So that's the action potential. It lights up. Okay, ding, like that. And then what happens in the next moment? This drawing is getting worse and worse every time. So this one, the sodium channels will close, potassium channels will open, right? All the potassium will leave. Then it will cause this to go all the way down to -90. But the next section, because it crossed the threshold a moment ago, so the sodium channel will open for this section, pushing it to +30. So now this section is undergoing the action potential. This section lights up. Okay, so ding, ding, right? And then the next one, because some sodium creeps forward, it will become -60 for this next section. My goodness, where are we? -60. The rest will still be -70. Okay, and then I think you kind of start to get this, I hope. Okay. This one will get reset by the sodium potassium pump back to -70, ready for another action. This one will get down to -90. This one will now experience the ding, okay? And then the next one will get pushed to -60 while the rest is -70. So if I keep on doing this and you follow the action potential, it basically just, goes down the axon, right? So like ding, 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 until it gets to the very end, until it gets to the axon terminal. And that's how the signal gets propagated. Okay, it flips the membrane potential a section at a time until it gets to the very end. All right. Now, just to add a little bit more of a twist, okay? Remember, the axon is actually myelinated, right? Okay. So you cannot exchange the ions. The sodium, potassium cannot come in and out if there's myelin. So what happens is, you know all that sodium coming in right here? Right? Some of it will go forward like that, and then it will trigger the sodium to come in, and then it will go forward, trigger to come in, forward, come in. So that caused the signal to essentially jump like this, right? And it's very fast. It's going to pass down the axon in no time. Are we okay? Seriously, any questions about any of these things? Okay, that's good. Here is a question for you to think about. Go ahead. I need to go to the bathroom quickly. Too much tea. You guys do this, Paul. I'll be right back. Okay, we have 3 responses, 5 now. Okay, just choose something. Even if you're not sure, choose F. Okay, so that I know you're there. Okay, looks like everyone is done. Okay, so this is obviously an application question, and it's a little bit confusing, but let's take a look at what happens. Okay, so hypokalemia, okay? Anything that's emia means is in the blood. So hypokalemia means low potassium. That can result in hyperpolarization of the membrane. All right, so what does that even mean, right? What that means is normally we are sitting at -70. Oh my goodness, it's so thick. Normally we're sitting at -70. Right? That's our resting potential, right? So that's the -70 that we normally sit at before we do the whole thing, right? But now the person has low potassium in the blood, and that results in hyperpolarization. So instead of sitting at -70, are we sitting higher or lower than -70? That's the first thing you have to ask yourself. Okay, so hyperpolarization means I'm further down. So I'm at like, whatever, -90 or something. Okay, so that's hyperpolarization. And if you're not sure how we know that, well, look at the graph, right? The graph we drew. Where's the graph? Right, like at the bottom, at the lowest point, it's hyperpolarization, right? And that's how we know it's minus 90. But that's not the point. The point is that it's lower than minus 70. Okay, so what happens, right? Let's take a look at these things. Which of the following will happen as a result? Okay, so number one, some sodium gated channels will spontaneously open. Well, that's not true, right? Because in order for the sodium channels to open, you need to be at minus 60, right? Okay, so we are further down from minus 60. Okay, it should be more difficult to open the sodium channels, right? So if there is another condition like hyperkalemia or something, then you'll be closer to the minus 60, right? Then the sodium channels will, spontaneously fire. Okay, and that's actually what happens if you have too much potassium in your blood. Okay, like your muscles start to have twitching and spasm. Okay, because the channels are like opening on their own. Right, so like you will be twitching a little bit. In fact, I've read an article that talks about like racehorse. They feed them high potassium in their diet just to like work them up, and so like their muscles are all like ready to go. And that's supposed to like enhance their performance, right? And again, it means that with a higher potassium in the blood, you're closer to the threshold, so like it's easier to fire those muscles. Does that make sense? Like why the first one is not? I hope so. Okay, resting potential is more positive than normal. So we talk about it is actually more negative than normal. A greater than normal stimulus is required to trigger action potential. That is true. That is the answer. Because we're setting at minus 90. Okay, and to trigger the action potential, we need to cross minus 60, right? So instead of like a difference of 10, we're now talking about a difference of 30, right? So that's why you need a greater than normal stimulus, right? So if you have hypokalemia, you might have muscle weakness. You try to move the muscle, but like it's like it takes so much effort to just to stimulate the muscle. It would feel like, okay, because of this big difference. Okay, uncontrollable muscle twitching, that's the opposite of what I said, right? It's high potassium. Threshold potential will not change, It's the threshold is always minus 60. Okay, it doesn't matter if you're high potassium, low potassium, it will not change. So the answer that you will choose is C. Any follow-up questions about why this is the way it is? All right then. Okay. This thing, I mean, I'm not gonna fill it in now. You should fill it in on your own if you haven't already, and I'll post the answer. You can check it. Let's talk about let's talk about some of these things, and then we will do the synaptic connection thing. First of all, any question about any requests for the long answer questions here? Any requests? Number 4. Okay. Yeah. So in order to do number 4, I actually have to talk about the neurotransmitter stuff as well. Anything else beside number 4? Okay. So let's go back to here. The drawings always get progressively bigger. I don't know why. Just make it smaller here so I can fit more stuff. You know what? Let me just take all this stuff and put it here so it will all be on the same page. There we go. Okay, so now let's talk about synaptic connections. Okay, so here we have one neuron. And then here we have another neuron. I hope you're still drawing with me, right? So we already know how the signal gets passed down, right? From the axon using the action potential that we spent the last hour talking about. And over here, we're going to be zooming in. Zooming in here. Zoom in. This was like a smart screen. Zoom in with your fingers. What you will see is this. Okay, you will have the axon terminal. Axon terminal of the presynaptic cell. Okay, so this is called a neuronal synapse. There are other types of synapse, like neuromuscular junction, but we'll just leave it like that. And then we will have this one. Right? This is the dendrite of the postsynaptic cell. Okay, so this is like neuron and neuron to neuron in this case. This space here, this is the synaptic cleft. It's important to know that the neurons are not actually touching themselves. Touching each other. There's a little bit of a gap in between. Okay. So the whole thing is a neuronal synapse. Okay. Now I want to remind you that it doesn't have to be a neuron to a neuron, right? Okay. A synapse could be between a neuron and an effector, right? It could be between a receptor and a sensory neuron, right? So you don't always have to have a dendrite for the postsynaptic cell. It could be a muscle, could be a gland, could be another organ. And that is important. So here we have some vesicles. Okay, and in the vesicles, we would have some neurotransmitters. And so these little blue dots, neurotransmitter, all right, anti. Right? They are chemical used for synaptic communication. Okay. Now listen carefully. I'm not going to write it. I don't have space. Some neurotransmitters. There are a couple of things you have to know about neurotransmitter. Okay. So number one. Some neurotransmitters are excitatory. Okay. It will remember those green arrows that we drew at the very beginning? Like here, right? So these green arrows, right? So some neurotransmitters are excitatory. They will promote these positive signals. Some neurotransmitters are inhibitory, right? They will promote the red signals. I don't want you to think about green as good and red as bad, okay? Because some people have this misconception. We actually need to have the inhibitory signals, right? In order for us to function properly. Think about it. When you're walking, right? When you lift your right leg up, you have to tell your left leg to not move, right? So that actually requires inhibitory signal to get there, right? Okay. Also, like if you think about in class, I don't know if you had this experience. So when you're in a physical classroom, sometimes like you're just so bored, right? Like teachers keep on talking, and you're just so bored. And in your mind, you're like, oh, I want to get out of here, right? But then you don't, right? You know that's not the appropriate thing to do at the setting. So you suppress certain action, right? So that is another example of where we need inhibitory signals. Okay. One more example. You're studying in the cafeteria or something, or at the library, You're so focused on your work that you zoned out the, background noises, the conversation from the next table, right? So all that zoning out, that's inhibitory signals. Okay, so it's not about good or bad. It's just about what you need at a specific moment. Okay, so all these, positive and negative signals, they are carried by neurotransmitters. Okay, so some neurotransmitters are excitatory, some neurotransmitters are inhibitory. Some, like dopamine, that we'll talk about, is both excitatory and inhibitory, depending on what region of the brain you're talking about. Okay, if you're talking about the reward pathway of the brain, then dopamine is excitatory. Okay, like you eat a cookie, feels good, I want to eat it again, right? That happiness, that good sensation, that's created by the dopamine. Okay, excitatory. Right? You might have heard people say dopamine is the happy hormone, right? Okay, you exercise, you feel good. That's the dopamine giving you that, excitatory effects. In another part of the brain, as covered in the lecture, there is something called the substantia nigra. Okay, the black substance in the brain. Okay, that part of the brain, dopamine has an inhibitory effect. It will suppress unwanted movements. Okay, so to recap, some neurotransmitters are purely excitatory. It doesn't matter which part of the brain it is in, it's going to be excitatory. Some are always inhibitory. Some are a little bit of both, right? Depending on which region of the brain it is released in. Got it? Okay. Another thing about neurotransmitters is that when you release it into the synaptic cleft, you will have like hundreds of other neurons in the vicinity, right? Okay, I'm just showing you like a one-to-one connection here, but in reality, right, there could be another neuron here. Okay, there could be another neuron here. Okay, so when the neurotransmitter is released, all of them will be exposed to the neurotransmitters. In order for a neuron to respond to a neurotransmitter, it must have the right receptor. Okay, so like, if these are blue neurotransmitters, so to speak, you will need to have a blue receptor. Okay, so over here you might have another neuron, but they only have like a red receptor. Then it will not respond to the blue neurotransmitters. Okay, you might have another one here, and this one has blue receptors, and this will react, and this will respond rather. This will respond to the blue neurotransmitters, but not that one. Okay. So the action potential will eventually arrives at the axon terminal, and at the axon terminal we have these calcium channels. Okay? These are voltage sensitive calcium channels. Okay, and what happens is when the signal arrives, the calcium channels will open, and then the calcium will go in. Remember, the inside is more negative than outside, right? So when you open the calcium channel, positive is attracted to negative. The calcium will come in, and then the calcium will bind to the little vesicles, and it will push them. It will push them towards the edge. Okay, so when it gets to the edge, it will fuse with the membrane, and you will release all the neurotransmitters. There's an animation that I posted in the video resource. Make sure you watch that if this is confusing. This is very low-tech animation, okay? The one that you will see on the YouTube is much nicer. Any questions so far? Like, seriously? No? Okay. Over here, instead of having a voltage sensitive channels, which basically means the channel is controlled by voltage, over here we have a chemically gated sodium channels. Okay, and just to reiterate, the sodium channels we learned like earlier, those are regulated by voltage, right? Like it opens at -60, closes at +30. These sodium channels are chemically gated. They are regulated by the presence of certain chemicals. In this case, the presence of the blue neurotransmitters. So when the blue neurotransmitter binds to the receptor, it will open them up. And then the sodium will come in. Sodium will go in. Okay, and if we have enough sodium going in, then it will push this -70 to -60, and then we will have an action potential. Okay, so that's how the signal goes from one neuron to the other. All right. Now, it's important to remove the neurotransmitters when we're done with it. Otherwise, you will keep on transmitting the same signal over and over again. If you've ever been to a concert, right? You might come back home at night and then you try to sleep. Your ear is still kind of like ringing a little bit, right? So that's like overstimulation, right? And then you might still have some lingering neurotransmitters around that keeps on stimulating the same pathway, even though the actual stimulus is gone, right? So we don't want to do that, right? Think about pain, right? Like you burned your hand, you're like, ow, get that initial burning sensation. You don't want that intensity to last like forever, right? That's got to be like really annoying, right? So you want to remove the neurotransmitters when you're done transmitting the signal. You can remove it by reabsorbing it. Okay, so take it back into little vesicles. You can remove them by enzymatically breaking it down. Okay, so that's basically what's happening. All right. Now, what was the question? Explain how abnormal neurotransmitter levels can lead to neurological order. Okay, so like I will talk about Parkinson as an example. Okay, so and again, all this is covered in the lecture. So in Parkinson disease, okay, we have low dopamine levels in the substantia nigra in the brain. Okay, and normally the dopamine in the substantia nigra will suppress unwanted motion. Like if I'm trying to write something, right? I don't want my hand to be like shaky and all that, right? I want to have precise control of my hand so I can suppress unwanted motion. That's why I need the dopamine, okay? So when you have low dopamine level and the substantia nigra, that will result in uncontrolled tremor. Okay, which is of course characteristics of Parkinson's disease, right? Okay. So how do we fix this problem? Well, if you don't have enough dopamine, you might be thinking about why don't we give them dopamine, right? Okay, but we have a problem. We cannot give them dopamine because dopamine will not cross the BBB. You have a blood-brain barrier that prevents things from crossing. Okay, so if you inject the dopamine or if you take it orally, none of those will make it to the brain, and it's not going to work. Okay, so instead, what happens is you give them like levodopa, which is not dopamine. This is a building block for dopamine, and it's designed to cross the BBB. So you take it orally, get crossed to the brain, into the brain, and then the glial cells, okay, will take these building blocks and make dopamine. Okay. So another thing is the clinical depression, right? I was talking about. Okay, so like there's another one called serotonin. If you have low serotonin, it has been linked to clinical depression. Okay, so what happens? This is what happens normally. Okay, like you have your neurotransmitters, you got your serotonin, and then you release them into the cleft, right? Okay, and then it exerts its effect. Right. So serotonin does a bunch of stuff, including regulation of your mood. Right. So what happens is in people with clinical depression, we don't have a lot of serotonin. Okay, we just have a little bit. Okay, so when they get released into the cleft, right, there's a receptor there for it. Okay, you don't get a lot of stimulation. Okay, so like they might bind to it, and then you would take them back up, right, into the vesicles, and then that would be it. So to fix this problem, okay, instead of giving them serotonin, you give them what's called like SSRI, okay, serotonin reuptake inhibitor. Okay, I forgot what the other S stands for, but I think that's what they're called, SSRI. So this thing, it will blocks the reuptake of serotonin. Okay, so even though you don't have a lot, that's okay. You release them into the cleft, and then the blocker would prevent them from being taken back. So what little you have, you will let them stay in the cleft for longer periods of time to continue to exert their effect. So you compensate the low level of serotonin by increasing the duration of their effect with the blocker. Okay, so like antidepressants, like Prozac, that's how they work. Okay, so I hope that answers 4 and 5. Okay, let's talk about the application question, and then we'll be done. First one. The first one is very long. Let's take a look here. Adenosine, as in like the adenosine triphosphate in ATP. Okay, adenosine acts as a neurotransmitter. When sufficient level of adenosine builds up, it slows neuronal activity and makes a person feel tired and sleepy, which kind of makes sense, right? Let's think about like ATP. You use ATP, it becomes like ADP and P, and then you can actually keep on doing it. It becomes like AMP and P. So when you build up a lot of adenosine, okay, in your system, it means that you're low in energy, and it makes you feel tired. Okay. Caffeine is a stimulant, and its effect is related to adenosine. Okay, based on the chemical structure shown here, explain how caffeine makes you feel energized. Okay, so I hope you can see that there is structural similarity between caffeine and adenosine. Yes, right. They both have these double ring here. All right, so let's take a look at our neuron. Okay, on the surface of a neuron. Right? I'm just going to zoom in on a dendrite. Okay, so here we have a receptor. Okay, we have some receptor. Okay, and so what happens is the adenosine. Okay, let's say the adenosine looks like this. Okay, the adenosine. Okay, so the adenosine, when you sufficiently build up the adenosine will basically bind to these receptors. Okay, and when the adenosine binds to the receptor, it creates like these signals, right? Like feeling sleepy, like that. Okay, so like more receptor, more adenosine. So now you drink some coffee, drink some Red Bull and whatnot. Okay, so the caffeine comes in, and then there is some structural similarity, right? So maybe the caffeine looks like this. Okay, so if you have enough caffeine in your system, the caffeine will basically compete with the adenosine, and it will block the receptor. Right? It will block the receptor. Right. So the adenosine will no longer be able to stimulate the receptor because it's blocked by the caffeine, and you don't feel tired anymore. Does that make sense? Anybody who drinks coffee here, like caffeine? Coffee, tea, Red Bull, Monsters? If you do, I want to ask you, do you feel the same effect as you first started drinking caffeine? Like at the beginning, if you drink 1 cup, you're like, oh, energized, go, go, go. But now, do you find yourself having to drink more and more in order to feel the same effect? Or like you cannot function without drinking one, even though it doesn't give you the same kick as before. Anybody had that experience and you know what I'm talking about? Yes. Yeah, you always need more, right? Okay. So here's why. Your brain always try to reestablish its initial state. Okay. Yeah, I get headaches too when I don't drink coffee. I quit already, but like at the beginning, I try to quit. Like I try to quit cold turkey, and I just get this massive headache, right? And couldn't even function. So I had to like gradually slow it down. But anyway, the point is, the brain is always trying to undo the changes that it's being imposed onto it. Okay, it's always trying to regain some kind of balance, if you would. Okay, so here's what happened. This is for real. Okay, if you drink caffeine, if you drink it 1 time, 2 times, no big deal. But if you keep on drinking it for like, half a year, couple of years, right? Then your brain will adapt. Okay, and you're like, okay. I mean, the brain is like, okay, you are flooding the system with caffeine and you're blocking the receptors. Okay. So, I don't like that. I want to regain the balance. I'm going to put more receptors on the surface. Okay. So that's what your brain will do. It will put more receptors on the surface. This is a neuron, right? Of course. Okay. So like there might be another dendrite here, and then it will have more receptors. So by putting more receptors, now the adenosine will be able to bind to all these extra receptors. Okay. And then it will trigger the sleepy feeling again. So you drink the same amount, it just doesn't have the same effect. Okay. And then you're like, okay, double, like a small coffee is not going to work. I'm going to go medium. So you get more caffeine, and now these caffeine will be able to compete again. And then you feel the effects again. All right. And then your brain will continue to fight it. It will put more and more receptor. Okay. So that's why like it feels like you don't really have the same effect anymore. Okay. So what I was trying to do, because I started drinking coffee in university. Okay. And it got me through university. And then I kept drinking into adulthood, right? And then eventually I feel like I was too dependent on it and I want to quit. So I quit cold turkey, right? Cold turkey means like I just cut it off completely. I thought I could do it, right? So that morning, I remember I had a math class at 8:30. I went to school, and I was standing in front of the classroom trying to do this calculation, and then my brain just like wouldn't work. I was just staring at it, and then I get this massive headache, and I was like, oh my God, this is not working, right? So physiologically, what is happening is you take away all these caffeine, you don't get your daily fix, so to speak. Sounds like an addiction. It really kind of is. And then now you free up all these receptors and all these adenosine that you've been building up. Now they just go like crazy, right? They just bind to all these available receptors, nothing competing for it. And then now you get this massive, massive stimulation that makes you feel like super tired. Okay. And that could trigger what we call like the withdrawal symptoms as well, right? The headaches, right? The sensitivity to light and so on and so forth. All right. So that is what is happening. Now, of course, for caffeine is okay. Like you can eventually quit, right? But as you know, right, there is an opioid crisis going on, and the same sort of stuff is happening with people who are addicted to opioids. Okay, and the withdrawal symptoms that they feel is much more intense. That's why it's not possible for them to just quit. Right? Some people say, "Oh, I don't understand why the people who uses drugs." They just don't quit, right? They can't. They physically can't quit it, right? The brain chemistry is going to be all messed up if they try to quit without a plan. And that's what these rehab centers are trying to do. They need to be able to taper it off and ease into it and not just quit like completely like that. Anyway, I hope that makes sense. Number 2. GABA is a neurotransmitter that causes chlorine to enter the neuron or potassium to leave the neuron. Based on this information, predict the effects of GABA on neuron and with a GABA receptor. Okay, so let's take a look at this. This is our, I'm just going to use this neuron. Okay, so normally resting potential is -70, right? You should know that by now. We're talking about it for the whole day. If there is GABA, GABA comes along. GABA, it will bind to the receptors. Okay, and what it will do is it will cause chlorine to enter the cell. Okay, so chlorine, of course, is negative. So if it enters the cell, what does it do to the -70? Does it make it more positive or does it make it more negative? That's the question you have to answer, right? Does it make it more positive or more negative? Well, hopefully the answer is more negative, right? More negative, because you are putting these chlorine into the cell and they carry negative, right? Okay. And then the GABA will also make the potassium leave. Okay, potassium will leave. So if potassium leaves the cell, it will also make it more negative. So together, the effects of potassium leaving, chlorine coming in, will make your cell hyperpolarized. Hyperpolarization, right? Which makes it more difficult to reach the threshold of -60, right? So based on this information, GABA is an inhibitory neurotransmitter. Okay. If you have something that makes the resting potential more negative, then it has inhibitory effect. If you have something that makes it more positive, then it has excitatory effect. All right, that is it. Very intense lesson, lots of information. Please study a little bit next week. Take some time off, of course, but don't forget you will have the quiz right afterwards, and it will only be on lecture 5. Okay. All right. Have a good weekend and enjoy your week off next week. I will see you the week after. Bye.